Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I am so happy to introduce our special guest on the show today. With us, we have Dr. Juan Domínguez-Bendala, who is Director of Stem Cell and Pancreatic Regeneration Programs at the Diabetes Research Institute at the University of Miami. Dr. Domínguez-Bendala's group recently published new research in Nature Communications that shows how THR-123, a small peptide, can trigger regeneration of insulin-producing beta cells in the pancreas, restoring normal blood sugar levels in a preclinical study, which essentially teaches the pancreas to heal itself. So very, very exciting research findings, and we're looking forward to learning more about it from the lead researcher on that study. And the findings chart a course toward a potential novel class of regenerative treatments for insulin-dependent diabetes. A bit more about Dr. Domínguez-Bendala. He earned a Bachelor of Science in Biological Sciences from the University of Seville and later served as a second lieutenant in the Spanish Air Force through a special program for top students. He also has a Master of Science in Applied Molecular Biology and Biotechnology from UCL, and then went on to do his PhD in gene targeting in embryonic stem cells at the Roslin Institute under Dr. Jim McWhir, a creator of Dolly the sheep. So that's very, very cool. Among the first to work on human embryonic stem cells, Dr. Domínguez-Bendala then joined the University of Miami's Diabetes Research Institute in 2001, where he has built his career from postdoc to faculty. He also chairs the university's Embryonic Stem Cell Research Oversight Committee as well. So to learn more about the very fascinating, very exciting work that Dr. Domínguez-Bendala is conducting and the new research findings from his work, let's turn to him now. Thank you so much for being on the show with us today.
Thank you, Ayesha. Thank you for the invitation.
And as I mentioned, you've had a very fascinating career, a very interesting background from Seville to UCL, the Roslin Institute, and now at the Diabetes Research Institute at the University of Miami, leading really cutting-edge research in the diabetes space. So I'd like to learn a bit more about that journey, if you could expand on that, and then what ultimately led you and inspired your focus on stem cell and pancreatic regeneration specifically?
Sure. Yeah. I've always loved biology, how, living things work. That's something that has appealed to me since I was a kid. And I think that knowledge for the sake of knowledge is worth pursuing on its own. Don't take me wrong. I think that basic research is amazing and much needed. But I always wanted that knowledge to be applied to human disease, to make an impact on people's lives. And, I was fortunate enough to come of age as a scientist, so to speak, at the same time, and in fact, in the same place where stem cells and the whole concept of regenerative medicine were being developed. So just make sure not to miss the train.
That is wonderful. And can you talk to us a bit more about, I think, your experience working with pioneers like Dr. Jim McWhir and, Dolly the sheep seems like it was a long time ago, but actually not that long time ago. I still remember as a kid hearing about it, but how was that experience like? And do you think that really charted the path towards really the kind of innovations and the innovative approaches that are that you're leading now in this space in diabetes research?
Yeah, that was an important time in the field of regenerative medicine. It was the first time that a mammal was cloned, and that opened up a lot of possibilities for therapeutic cloning. That was a concept that was in vogue at the time. The idea was to create cells that were identical to the donors and that you could use to regenerate tissues that wouldn't be rejected. And that was a big thing at the time. It really open up all these different possibilities. Now it kind of got outdated in terms of new techniques appear that allow you to do the same thing, like cell reprogramming, with Yamanaka getting the Nobel Prize at the beginning of the 2000s to do the same thing, but without the need for embryos. But it was a very active time, and the whole stem cell field appeared there. I think it was in the year 98 that the first embryonic stem cells were developed, and we had access to those cells at that time, at the same time, at the University of Edinburgh in Scotland. So all those experiences obviously shaped me as a scientist, and I recognized very soon that all that knowledge could be applied to diseases like type 1 diabetes, which is what the Diabetes Research Institute was all about. Because when you have diabetes, you need to restore the beta cells that you no longer have because of the autoimmune attack. So stem cells were a way for us to get more of those beta cells. So that was a very impactful time in my life, and I really am thankful for the opportunity I was given at the time as a student to be next to the people who shaped the field for decades to come.
Indeed. And have essentially made things like cloning very mainstream, at least in the research arena, for sure. I think it's in any lab, in molecular biology lab you go, that is kind of a very conventional technique now. And so to think that was so groundbreaking, and that's, really, really cool to hear about the evolution and how far we've come, and what it took to get to that and to get to where we are today. And so in terms of the diabetes space, really, what in your opinion do you think are really the unmet needs? I think they're pretty obvious, but I feel like a lot of people overlook the fact that diabetes is treated like a chronic disease, which it is, but it can be managed pretty well now. People, it's not life-threatening per se. But so what do you think the perceptions are right now and today, and in terms of what the unmet needs are that people may not really pay attention to, but that are very pressing and urgent?
Right. So there are many types of diabetes, and the diabetes that most people are familiar with is the type 2 diabetes, which is a type of condition where your stem cell, your beta cells in the pancreas are still making insulin, but the cells of the body don't recognize that insulin very efficiently. And then you develop something called insulin resistance. And eventually, because the cells have to work more to get to the same point, they get exhausted and they may die. So that's another complication. It becomes a beta cell disease at that point. But still, type 2 diabetes is a disease that, as you said before, it can generally be managed before it becomes a life-threatening disease. Even though I'm not saying it's something that you should just tiptoe around the fact that it's a chronic disease, and obviously you need to be very careful about that. But I think that the more pressing, urgency in the field is about type 1 diabetes, because it's a completely different disease in its origin. It's an autoimmune disease. It used to be called juvenile diabetes because it affects children and young adults disproportionately. So in this case, what you have is the autoimmune destruction of the beta cells. Your immune system makes a mistake and thinks that the cells that are making insulin in your body are foreign and destroys them. And when you don't have any more beta cells, you don't have insulin. And without insulin, you cannot survive because the glucose that you take, that's the fuel for your cells, cannot get into the cells. You have to use alternative fuels, and those are fats and other things, which sounds good in principle, but it's really bad. If that's the only fuel that your cells have, many bad things happen. So in short, you need insulin to survive. Without insulin, people with type 1 diabetes would die. And insulin doesn't replace islets. No matter how good a control you have of your diabetes using insulin, you have your ups and lows over years that bring about many, many complications over time, including blindness, kidney failure. So it's a really devastating disease and a burden on the people and their families that have it. So that's the disease that we are aiming to cure at the Diabetes Research Institute. And in order to cure anything that comes with the word cure, for type 1 diabetes has to involve two things. One of them is to stop autoimmunity, which is the underlying cause of the disease. And the other is to bring back the cells that were destroyed by the immune system. If you don't do any of those two things, then you don't have a cure. You may restore a normal immune function in the patient, but if you don't have any more beta cells, you're still diabetic. And the other way around, you can bring back the beta cells, but if they get rejected, then we are in the same place. So um, to address this, we have been pursuing, as an institute and in general the field, very aggressively, we've been pursuing the approach of islet transplantation, which is an approach by which you can take the beta cells or the islets that come from a cadaveric donor, from a pancreas, from a donor, and you can transplant those islets into a patient. And that works beautifully. The only problem is that you only have a handful of people or pancreas available for transplant every year, and you have millions of people with type 1 diabetes. So that doesn't work very well because of the scarcity of organs for transplantation, and also because of the need for immune suppression. These are tissues that come from somebody else, so they will need to be protected in order for them to graft and survive and thrive in your body. So those are the two main limitations. Now we are working with stem cells. They are taking the place of islets. We can differentiate the stem cells in vitro, and we can grow them until we have enough. And we coax them into becoming beta cells, and we are transplanting them into patients. We are doing clinical trials on this. So that's in itself something that has been a major advance in the last 5 to 10 years. But there's another approach that we are pursuing in my team, and we've been doing this for quite some time already, and is to try to induce the pancreas to regenerate its own beta cells. And this is not a new concept. It's been around for nearly 100 years, but we've been very busy trying to identify the cells in the pancreas that can respond to specific growth factors. And one of them that we discovered was BMP7. We didn't discover the growth factor. We discovered that the growth factor, BMP7, stimulates those cells in the pancreas, those stem cells in the pancreas that can give rise to new islets. And this BMP7 molecule, this growth factor, again, is circulating in your blood and in mind. It's made in the kidneys and in the bones. And it's like fuel for stem cells. So every time there's a need for repair and regeneration in the body, these circulating factors help in their regeneration. So what we did was simple in at face value and very complicated when it comes to the details, but we made animals diabetic and we treated those mice with a molecule that is similar to BMP7, which is called THR123. It's a shorter version of the protein, BMP7. It's simpler to synthesize. It's also cheaper. It's easier to take to the clinic as well because other THR molecules have been already in clinical trials. And what we did was to treat diabetic mice with THR123, and we saw that the islets grew back. The glycemic control of the animals came back. So we restored normal glycemia in those animals. And when we look inside the pancreas of those animals, we saw lots of islets growing back to the way they were before we made them diabetic. So that was quite striking, and that's the gist of the discovery that we are reporting this year in this paper.
That's very, very fascinating, very, very exciting indeed, to be able to really regenerate those islet cells using this factor THR123. And so how would this work therapeutically compared to insulin treatment, traditional insulin-based treatments such as injections, or as you mentioned, the other option would be islet cell transplantation. So would the idea be to give patients THR123 continuously, or would it be for a certain time? So what would it entail clinically?
Right, that's a very good question. I mean, first you mentioned insulin injections and how this is different. Insulin injections have to be for life. It's a chronic thing that you have to endure. So when you do islet transplantation, that's already doing something about it. You're replacing the cells that make insulin by means of a transplant. And you can do the same with the stem cells. But if you ask me, transplantation, and this is my personal opinion, is still kind of a brute force approach. You're putting cells against their will in a place that is not where nature intended. It's not in the pancreas where you're putting the cells, and where they're being attacked by the immune system. So basically, you need to protect them in all kinds of ways to prevent their destruction. So we think that inducing the regeneration in the pancreas, which is where those cells are supposed to be, is a much more holistic and comprehensive way to approach the problem. And I think that you asked specifically about the way that this will look like in the clinic. If this would be a chronic kind of administration or something that you do at the beginning and then you forget about. We think it's the latter. We think, and based on our data, what we are doing is to stimulate the pool of progenitor cells in the pancreas to produce new islets. In mice, the treatment takes about 2 to 3 weeks. We can extend the treatment for up to a month, and then we got the almost the full complement of beta cells back. So it's a very powerful approach. We are speeding up the process by which new beta cells are created. And we don't discard the possibility that down the line, the patient may need a booster, kind of, another, stimulation of those, residual progenitor cells if you need more. But in our view, that would be a one-time kind of deal. You do this for a few weeks, and then you regrow the beta cells, and then you don't have to do anything else.
Mm-hmm. So really, with that curative potential there, just with the one time, or as you said, maybe a booster, I think that's very, very powerful and very, very, as you said, like transplantation comes with so many complications, graft versus host disease and all of those rejection and all of that kind of thing. And here, really, you're teaching the pancreas to heal itself is what I kind of read about this approach. So I think that's very, very powerful indeed. So yeah, the preclinical data, as you mentioned, sounds very, very exciting. And so how far are you to taking this in clinical trials in patients? How far are you from that?
Right. So we still need to do preclinical work to ensure efficacy and safety. Safety, we are less concerned because these BMPs in general have been used clinically for decades. For instance, you use BMP7 every time you have a spinal surgery when they have to fuse vertebrae. They use BMP7 to bring the two bones together and regrow new bone there. And it's been very, very safe. And other THR molecules have already been used in clinical trials. And they've been given systemically. And that's the beauty of the approach, is that it doesn't work everywhere, even though you're getting it all over the place in your body. It's going everywhere.
But so it's targeted.
It's not targeted. It goes everywhere, but it only works where it is needed. And I'm going to tell you why. Well, we think that's happening. So we have BMP receptors in every cell of the body, but not at the same level. So when you have an injury in any tissue, you upregulate these receptors, and it's like kind of an SOS signal. And these circulating BMPs, they will bind to those cells with more, affinities, because they have more receptors. And that's why when we give BMP7 to healthy mice, non-diabetic mice, or THR-123, nothing happens. But when you have an injury in the pancreas, like we induce when you make those animals diabetic, that's when we see the effect. It's not targeted in the sense that we are driving it to the pancreas, but it's targeted because
but the injury's already there, right?
Right. So the efficacy, we are testing and being able to reproduce it in higher animal models. We want to do also non-human primates as well. We hope to be able to do all this boring preclinical work for the FDA within the next 3 to 5 years, perhaps. And at that time, we'll be ready to present this. It is, because the discovery part, which is the exciting one, it's done, which is great news. Now you have to do lots of tedious and boring work to reproduce it and to make it effective, and the pharmacology, pharmacokinetics, biodistribution, all those things. But it's good news that we are doing that, because it means that we are already on our way to the clinic. And importantly, and that's something I didn't mention before, and many people say this many times to me, this was work done in mice. So you have to keep into consideration that we have cured diabetes in mice in many, many different ways. It's not so difficult to cure diabetes in mice. And then when we have tried to apply those things to humans, we haven't been successful. So you have to take this with a grain of salt. This works in mice, and we need to make sure that it also works in humans. The good thing that we have going for us right now is that this is only the the last of a series of experiments that we've been doing over the last 10 years in many different models. And one of them, which we have pioneered, is the use of human pancreatic slices. So these are slices of the real living human pancreas that we can keep alive and culture for up to 2 weeks. And we've got slices of the pancreas of donors who had type 1 diabetes at the time of their passing. And even 2 weeks after we got that tissue, that little piece of the pancreas is still alive in our lab. So we have the THR-123 and all these THR molecules in the MP7, and we have seen that even in that pancreas from a type 1 diabetic donor, we see a regeneration happening. So it doesn't get any closer to a real life clinical situation than getting the human tissue from a donor who had the disease and see regeneration happen before your very eyes. So that's something that we have. It's an advantage of our system that we have tested it across many
Different
models, and it's
models.
not
Yeah.
just yeah, yeah.
Wow, that's so exciting, and that's so that very very hopeful, right? That's I mean to be able to see those results, not just in animal models, as you said, where the physiology is obviously very different from the human setting, but to have a model like a pancreatic, specimen, a slice, and then seeing those results in that as well. That's super exciting indeed. And in terms of, I would ask about, would this approach, could this be applied to other disease settings, do you envision, outside of type 1 diabetes? Just when looking at the whole field of regenerative medicine, and then what conditions you could potentially treat with this type of approach?
Well, that's a really interesting notion. My hands are full as they are with type 1 diabetes work, so
Don't
I don't
we
pretend
won't
to.
ask you to today take on any more.
No, no, but it's interesting because of what something that I mentioned before. I told you that these BMPs are circulating in your body, and they're kind of looking for tissues that are in danger or distress, and they help the regeneration. Our model is one where we induce an injury in the pancreas, and we see a regeneration in there. I bet that if we were to use a model in which the injury is in the liver or elsewhere, the same may apply. And again, I wish I had the time or the people to do all those things. But I know for a fact that THR-123, the same one that we use for regrowing beta cells, has already been used to restore or to correct fibrosis in the kidney, for instance, which is something that may come about when you have diabetes for many years, but it's not diabetes, it's something else. So it has the potential to be usable in many other applications for sure. That's a really nice notion to explore.
Absolutely. And in terms of the field of stem cells, regenerative medicine, how do you see it evolving in the upcoming years? Actually, how have you seen it evolve in the past decade, 2 decades or so? And where do you see it headed? Is it going to be I think when we think about, is it going to be cell-based? Is it going to be tissue-based? Is it going to be molecules coming in to regenerate tissue cells as THR-123, or a combination of approaches? What trends do you think we're going to see, and where is the field headed?
Yeah, that's a good question, and it helps to have been working on this for 20 years since
Yes.
I
So
joined
you're
the
the perfect
DRI in the year
person to
2000.
ask. Yeah.
Well, yeah, I mean, because I was the first one to bring stem cells and human embryo stem cells to the state of Florida back in
Oh, wow.
2001. So I've seen the whole field evolve, and at that
Evolve,
time
yeah.
we were going to cure all diseases. Every condition was going to be cured with stem cells,
Yes,
and of course,
for sure. I remember. Yeah.
yeah, we've had quite a bit of a reality check, but still, it's a lot of expectations and hype at the beginning. Then it goes down when you realize that things are much more difficult than you envision. But then it picks up speed again, and I think that I've seen all happening within the past 2 decades. So at the beginning, when we started this, we thought it was going to be a matter of just a couple of years. We differentiate stem cells into beta cells, we transplant them, and we're done. And it has taken nearly 20 years to do so, and for many reasons. And I can talk for a very long time about this, but when you have stem cells, these kind of pluripotent stem cells, like embryonic stem cells, um, it's not the same to make them into neurons or beta cells. For instance, just to mention two cells that you may want for therapeutic purposes. They readily differentiate into neurons and CNS kind of tissue. They don't like to become the pancreas. They hate to become pancreas. You need to push them really hard for them to make the right decisions along the way so that they become a human pancreas kind of. So it has taken the field collectively a long time to figure out the conditions to make them go in this direction. And also at the beginning, we saw there was so many different stem cells. You can get them from the fat, and you can get them from the teeth, and you can get them
Right.
obviously from the umbilical cord, and not all the stem cells are created the same. And at the beginning, everybody thought, oh, well, I can use umbilical cord blood cells to recreate islets, and people spend a lot of time trying to do that. Even I would spend some time, and it's not the right building block. So all these things, I mean, it's been a process of trial and error, and it's good. That's the way that science works. You see something works, if it doesn't, then you take another path. So if you keep doing that over the years, and you do it for long enough, like we've been doing it for nearly 2 decades, you see that your path ahead is narrower and narrower. You have discarded many other options that you saw that were leading to dead ends, and that makes us very confident that where we are heading is in the right direction. Where you see this happening, we are going to see stem cell therapies like transplantation of stem cell derived islets. We're going to see that in the next few years. It's happening already in clinical trials, right? And there has been a major development, because when we had to rely on islets, you simply cannot get enough. With the stem cells, you have an unlimited supply of those. So, of course, you need to solve the problem of immune suppression, because you don't want to treat the recipient with immune suppressants that obviously have lots of complications. So I think that's going to happen. The field is pushing very hard towards the notion of putting those cells inside a device, an immune isolation device, and that's a way to protect them from the immune system without the need for immune suppression. But it hasn't worked. So we failed in 2 clinical trials. The immune system is very smart, and even though these materials that we use to protect the cells are supposed to be inert and not immunogenic, there's something weird that the body recognizes, and eventually these cells die. So, but I think that progress is being made little by little. If the first 2 clinical trials didn't work, there will be a third and then a fourth. Eventually, these things will be made to work. When they do, I am personally skeptical that they will last for a lifetime. But let's imagine that they last for 6 months, right? Before you need to replace them. You're transplanting these things under the skin. It's not like you're putting them in the liver or in the pancreas. So it's a very accessible place. So if a patient of type 1 diabetes has to come back to the clinic every 6 months for a transplant under the skin in a device, and that person forgets about diabetes for 6 months, that's not a cure, but it's very close to what people would consider a cure. In the sense that it's what they call a functional cure. If you can forget about your insulin, that's what people with type 1 diabetes want. They want a break from insulin. They want to go to bed every day without the fear of getting a hypoglycemia during the night. So this thing will change the state of the art, and they are happening right now. So I think that those approaches where you're transplanting cells, those are going to be the bridge towards the cure. I think that what we're in terms of regrowing new beta cells in the pancreas is going to be much closer to what nature, ended with. And that will be closer to a cure. But by that time, I think that we'll be at a point where we are going to see transplants that may last a few months, and people will be very happy having to come out to the clinic every few months because they have forgotten about diabetes for that long.
Indeed, I think it's really eye-opening to kind of see how far the field has come, and really this approach, this in vivo approach, I think is very, very powerful. I think With cell-based therapies, it's always been the challenge of transplanting, either taking cells out from a patient, engineering them in a way when we talk about gene therapies, and then transplanting them back. And I think now, even on that side of things, gene therapies and cell-based gene therapies, we're also seeing the same ideas where people are looking more at how can we do things in vivo, in the body. So I think that seems to be kind of where these spaces are heading. And it also reminds me of gene therapy, how it was, proof of concept for so long. I remember reading about it when I was in school. And then finally, only in the last several years have we really seen gene therapies start to take off in terms of getting to clinic and to getting those approvals. So very fascinating. And to see the same thing happening here as well. In regenerative medicine, it's very exciting. And so in terms of, you've worked with pioneers and you are now a pioneer yourself. What advice would you have for people looking to get into research today? What insights can you give them?
Well, that's a difficult one, but I think that making it in science is, of course, it's a matter of talent and perseverance, and they will tell you that you had to persevere, and that's absolutely correct. That's right. You cannot abandon things at the first time that they don't work the right way. At the same time, you need to be aware and cognizant of when to cut your losses, because sometimes you may spend 10 years doing something that obviously is not working. So you need to know when to stop as well. But I think at the end of the day, leaving aside those two things that you can control, it's also a matter of luck. And a lot of it has to do with being in the right place and at the right time. And that's those things. I mean, sometimes you cannot control. Sometimes you can try to be in that, place. But my advice would be always to keep your eyes open to recognizing an opportunity when it presents itself, and don't let your plans stand in the way of a new development or a new opportunity that may take your career in another direction, because you may have the best laid plans and then life has something different in store for you. So be right, but be open to changing your plans. Sometimes your plans are cast in stone, and that limits you, right? So be open to the possibility and go with the opportunity if it presents itself. That would be my advice.
Wonderful. Also, I wanted to ask you about academia versus industry. We're seeing a lot more collaboration and partnerships happening between the two spaces. But when we, traditionally think about drug discovery and where medicines come from is the, big pharma or biotechs. But it's really great to see that, research institutions are, traditionally and historically where the discoveries have been and are still really being made, and industry even recognizes that. Do you think there is more collaboration happening between academia and industry? You also have some, academic centers and places where they encourage entrepreneurship and, collaborating with industry. How important are those relationships? Are they important, or is academia where it is? Is it okay just being an entity on its own, or do you think those collaborations are important?
No, it's super important. We've had and we keep having collaboration with industry, because even though many discoveries are made in academia compared to industry, but industry has the ability to take those discoveries all the way to the patient. For instance, in our case, we have this compound, this peptide THR-123, and we want to take it to the clinic. We can do all the preclinical work through standard, academic means like grants and such. But when it comes to doing a clinical trial, and particularly the big clinical trials, I'm not talking about small phase one, which perhaps we can do in academia, but phase two and phase three, you need a partnership with industry. So industry will many times be able to recognize the potential of any approach, and they will want to partake at the very beginning, right? So they will invest in your research, they will sponsor your research, and then, of course, you will co-develop things together. It's super important. And in the field of diabetes, even more so. And it's funny because many people have been asking me over the years, well, is Big Pharma really interested in a cure for type 1 diabetes? Because after all, they're making their money from insulin, right? So it's not in their interest. Well, Big Pharma has invested billions over the past 10 years in curative, so to speak, approaches, in stem cell therapies. Why? For the simple reason that they see that this is going to happen. And when it happens, and this is a very selfish, so to speak, kind
They don't want to miss out, right?
understanding, they want to miss out. They want to be the ones who sell it to you, right? So, but
Right.
it's a good thing for the field to see that they are investing so heavily because they see that insulin is not in the future. It's gonna be something else, and they want to be part of that. So we are seeing a lot of investment from industry at this stage for all these approaches.
That's excellent. And yeah. Also, in terms of looking ahead in the next couple of years, in the next year and in the next couple of years, what are you and your team looking forward to in terms of any, well, you did mention milestones and the work that you're continuing, of course, to get the preclinical work done and then get to the clinical stage. But what are some milestones that you're looking forward to coming up?
Yeah, I mean, it's what I said is basically the pipeline that we have to take this to clinical trials, but it doesn't end there. It doesn't end in the clinical trial because many clinical trials fail. So I'd be happy when we see promising results in the clinic, when those clinical trials are telling us that we have regeneration in people and we are making a difference in the lives of people living with type 1 diabetes. That's what we have done today is to publish a paper. That's great. I mean, congratulations. But that's not going to change anything. We're at the beginning of a race that doesn't end here or in a clinical trial. It ends when you have a product that you can have in the market or an approach, a strategy that changes the lives of patients. So that's our objective. We have that down the line. It's what we're aiming at. So, yeah.
Wonderful. Dr. Domínguez-Bendala, thank you so much for your time today. It was so wonderful speaking to you. And thank you so much for the very important, the very wonderful work that you're leading in the field. So thank you so much for all that you do. And thank you for your time once again.
Thank you so much for having me. It's been a pleasure.
Likewise. That's the end of this episode of the Xtalks Life Science Podcast. We hope you liked today's show. Please don't forget to rate, review, and subscribe, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.